Hi friends, today we are going to be talking about meiosis. Make sure that you are taking notes as we go and follow all the instructions on Schoology. All right, so first off, this term meiosis does not mean the same thing as mitosis. All right, they're both words that people tend to get confused. They start with M, they both deal a little bit with cell division. They are not the same thing. So it's really important that you recognize we're learning about meiosis, not mitosis. So mitosis is the making of somatic cells, which are non-sex cells. We're going to talk a lot more about mitosis in a later unit. Today, though, we are talking about meiosis or meiosis. I say meiosis. You can say meiosis or meiosis. Doesn't really matter, right? That's the making of sex cells. Okay, so different than mitosis. So we're making sex cells. So before we get into the steps of meiosis, we should talk about some vocabulary first. Gamete is a sex cell. So this is a term we've heard before, but just to refresh your memory, this is a sperm or an egg cell in humans, right? Gametes are sex cells. Homologous chromosome. This is a word we're going to hear a lot in this video. So a homologous chromosome are the two chromosomes that are the same number, right? So for example, you have a chromosome number 17, right? You have one from mom and you have one from dad. So homologous chromosomes are those two number 17s. Right, so they have the same genes on them. They're just different because they're, inherited from a different parent. But the homologous chromosome are the matching chromosomes. So number 3 and number 3 are homologous. Your X and your other X, if you're female, are homologous. Those are your homologous chromosomes. And here's a good picture of homologous chromosomes so you can kind of see what they look like. So that picture on the right there, that's called a karyotype, and you can see the chromosomes are lined up with their homologous pair. This person is male though, so you can see down here they have one X and one Y. They don't have a pair, right? But the others are all homologous. Tetrad is what we call when those homologous pairs are duplicated and standing with their homologous chromosome. So duplicated is after replication. So in replication, the DNA will copy itself. And then if it's standing with its homologous chromosome, it's a tetrad. So here you can see in the picture. So one side of this is one chromosome, right? It's been copied during replication. And then this is our other chromosome, right? So this is like number 10. This is an identical copy. This is your other number 10 from dad, right? This is the identical copy. So together they make up the tetrad. And this is just a little picture to show you replication, right? So how we get an X. So this is like your normal state. Your chromosomes are not replicated. They're just hanging out by themselves, right? So this would be like your number 10 from mom, your number 10 from dad. After replication, right, then you have these sister chromatids. They're identical that are attached, right? And so then together, this would make the tetrad. Diploid is another word we need to know. So diploid means two sets. So any cells that are diploid cells are cells that contain two sets of chromosomes, right? So all of your somatic cells, your normal cells are diploid, right? Because you have a set from mom and you have a set from dad. So we call that 2N, where N is the number of chromosomes, right? So humans are 2N, two pairs of 23 chromosomes. Haploid means one set. So those are cells that only contain one set of chromosomes. These are your gametes. Your sex cells are haploid cells because they only have one set of chromosomes. And we'll see how that happens today during meiosis. An allele, this is another word we've heard quite a bit, but to refresh your memory, an allele, remember, is a form of a gene. So plants have a gene that tells them if they're going to be tall or short, right? One allele makes a tall plant, one allele makes a short plant. So the forms of the gene. All right, so when we talk about humans, let's kind of review here. Remember, somatic cells, again, are diploid. They have 46 total chromosomes because they have two sets of 23. And your sex cells, or your gametes, are haploid. They only have 23 chromosomes. And we learned in Standard 1 that those chromosomes are randomly arranged, right? It's not like all of mom's gets on one side, all of dad's on the other, right? They're randomly mixed up in each of your gametes. Chromosome number, though, does not determine the intelligence, doesn't determine the complexity of an organism. So just to give you like a little bit of knowledge on this. Humans, again, we have 23 chromosomes. Flies have 4. Dogs have 39. Horses have 32. A potato has 24. Rice has 12. And onions have 8. So you can see that chromosome number doesn't really mean much, right? I think we would all agree that a potato is not more complex than humans, but they have more chromosomes than us. So chromosome number does not tell you anything about complexity, about intelligence, anything like that. So just kind of keep that in mind. All right, let's get into meiosis now, right? So meiosis is a process that's going to separate homologous chromosomes, resulting in a haploid cell. Okay, so we're going to start with a diploid cell, 2 sets of chromosomes. We're going to separate those chromosomes and then separate those cells so we get haploid cells, right? So meiosis is the making of sex cells. That's what we're about to do. And again, haploid gametes are going to be produced from diploid cells. So we're going to start with a cell that has 2 sets of chromosomes, and by the end we're going to make cells with only 1, right? So we're doing this process of making gametes. All right, so keep in mind, before meiosis can happen, replication has to happen. And replication is a word we've heard a couple times, right? Remember, that means the DNA is copying itself. So think of that one chromosome, right? It's going to make its identical twin, which is called a sister chromatid, right? That has to happen before meiosis can happen. There are 2 rounds of division in meiosis. So we're going to talk about meiosis 1 and meiosis 2. There's 2 parts to it. All right, meiosis 1. So the result of meiosis 1 is the homologous chromosomes are going to be separated. So once we get through all 4 steps of meiosis 1, that's what you should see at the very end. All right, the first step of meiosis 1 is called prophase 1. So there's a couple things that happen in prophase. 1, your chromosome is going to line up with the homologous pair and it's going to form a tetrad, right? So again, your number 14 from mom is going to find 14 from dad, and they're going to stand together. They're going to make a tetrad. Spindle fibers start to form. So in this picture, these little orange guys are spindle fibers. You'll see them start to reach out across the cell, and they're going to try to be connecting with these chromosomes, right? So they're reaching out towards the chromosomes. The nuclear envelope breaks down. The nuclear envelope is just this envelope or membrane around the nucleus, and you can see in the picture it's starting to break down, right? So that's the chromosomes, can get out of the nucleus so that we can divide them. And then something called crossing over occurs. So crossing over is when your homologous pairs are going to exchange a little bit of their chromosome. So if you look in the picture, right, let's say blues from dad, reds from mom. Let's say this is number 8. Okay, so your number 8 chromosomes are standing together as a tetrad, right? And they're going to crisscross here, and they're switching a little bit of their chromosome, right? So now you have this side, this sister chromatid is still all from dad. This sister chromatid is still all from mom. But these other 2 now, it's mostly dad, a little bit of mom. Mostly mom, a little bit of dad. So if these were genes, right, genes A, B, and C, we now have a new combination here that neither parents had before. So crossing over gives us genetic diversity, and that's really important. Think back to our evolution unit, right? In order to survive, if the environment changes with natural selection, you need to have genetic differences, right? So that way something can survive. One of us is going to have the right traits to survive. All right, the next step is called metaphase 1. So in metaphase 1, those chromosomes are now lined up on this metaphase plate, which is the middle of the cell. And you can see these little spindle fibers have attached, and that's what pulls those chromosomes to the middle of the cell. So the spindle fibers are the things that are directing the flow of those chromosomes. And again, they're going to line up here, and you can see that they're lined up as homologous pairs, right? So number 1 and number 1 are together, number 2 and number 2 are together, number 3 and number 3 are together, right? And you can see here, right, crossing over has occurred. So in some sources, you'll see that crossing over, they put it in prophase 1. In some, they put it in metaphase 1. It kind of happens a little bit in both, but you can definitely see here that crossing over has occurred. The next step is called anaphase 1. So in anaphase 1, what's going to happen is these spindle fibers shorten, and what that does is it pulls apart those homologous pairs, right? So again, on the metaphase plate, if we had chromosome number 1 and chromosome number 1 here, right, the homologous pair, then in anaphase, they're going to be pulled apart to opposite sides of the cell, right? Because remember, we're trying to separate them. And then the last step is called telophase 1, and it combines with cytokinesis. So in telophase 1, it's like the last part of meiosis 1, These chromosomes are completely separated from each other. Those spindle fibers are going to start to break down. You start to have a little bit of a nuclear envelope form. And then cytokinesis is division of the cell. So that cell is going to pinch in half and make 2 cells. That's meiosis 1. All right, and so we went from a cell that was a cell that was diploid, right, that had 2 sets of chromosomes, from a cell that is now haploid. So these new cells have half as many chromosomes as the parent cell had. All right, now we are on to meiosis 2. All right, so meiosis 1, now meiosis 2, and in meiosis 2, what's going to happen is those sister chromatids are going to separate. So remember, the sister chromatid was that identical copy, right? So if this is your number 7 for mom, right, during replication, that gets copied. You have 2 number 7s for mom. They're identical and hooked together, right? So in meiosis 2, we're going to separate those sister chromatids. And it's happening in 2 cells because remember, at the end of meiosis 1, we made 2 cells. This is going to happen in both. All right, the first step is called prophase 2. So kind of the same idea as prophase 1. Your spindle fibers are going to start to form and your nuclear envelope is breaking down. Metaphase 2, your chromosomes line up in the metaphase plate. The big difference you should notice is that now they're standing alone, right? In metaphase 1, they were with a homologous pair. Now in metaphase 2, they're all alone, right? So it's just a nice, neat line down the middle of your cell, just by themselves, right? Because if this is your number 1 from mom, number 1 from dad's in a whole different cell, right? They can't be in homologous pairs because they're separated. Next, you have anaphase 2. So in anaphase 2, those spindle fibers shorten, right? And those sister chromatids are going to pull apart, right? So you're separating out those identical copies of the chromosome. And then telophase 2 and cytokinesis. So telophase 2, your spindle fibers break down. You have nuclear envelopes forming, and then your cells divide. And the result of this is going to be 4 daughter cells, okay? Because both of those cells from meiosis 1 now divide again. So at the end of meiosis 2, we have 4 daughter cells that are haploid. So again, they have half as many chromosomes as the original cell had, right? So if it's a human, the original cell had 46. Now after meiosis, you have 4 cells, each with 23 chromosomes. Okay, so again, it's going to give us 4 gametes or 4 sex cells. And interestingly enough, in women, only 1 of those cells are actually going to be functioning, right? So egg cells are pretty big, so 1 of them is going to get everything it needs. The other 3 are just something called polar bodies. They just get broken down by the cells. In males, all 4 will go on to become sperm cells. It's kind of an interesting tidbit, right? Because sperm cells are so much smaller than an egg cell. They don't need as much, so you can make 4 viable gametes in this process. All right, so make sure that you finish the assignment. So go back to Schoology and look at the instructions for what comes next, and we will go over this in class tomorrow. Thanks, guys.